All right, you guys. So our last set of notes here on chapter 10 just covers gene mutations. Okay, so I'll just recap at the end of this when we talked about chromosomal mutations and just emphasize the importance of not confusing the two. Okay, so gene mutations are essentially changes in the nucleotide sequence, so changes in the genetic code of which we were talking about the nucleotide sequences of our A, T, C's, and C's and G's, right? And so a simple example shown at the right here is here we have a DNA section, a section of DNA coding for the protein hemoglobin, which carries oxygen in our red blood cells. And we've got this nucleotide sequence here showing the code CTT, which would correspond to the codons, right? So remember the mRNA codons of GAA. So C pairs with G, T pairs with A, right? And this would result in the amino acid at this position called glutamate, which is shown here. A simple mutation in which the T in the middle of the genetic code is substituted with an A leads to a codon GUA, which changes this glutamate to the amino acid valine. And this one simple change, this is one amino acid in thousands of amino acids that make up hemoglobin, but this one simple change converts the shape of the hemoglobin, and it causes the shape of the red blood cell to change into that sickle cell format. If you remember back to chapter 9, I looked at this. I'm trying to think of where I showed that to you, but I think back here, blood type, sickle cell, there we go. So if you look at the shape of the red blood cell, very different from what you're used to looking at if you remember the cancer lab and looking at red blood cells, very round, circular in shape. I'll put up an image of these and I'll restart the video so you can see that. So here you can see on the right-hand side, we've got normal red blood cells. And then in this individual, we have sickling cells, which have these kind of sickle-like shapes, kind of curved. There's different cartoon pictorials of them, etc. Now we can imagine in terms of how this affects blood flow. So one, it affects the delivery of oxygen. And so people with sickle cell anemia typically have very low energy, fatigue, tired, sore muscles, lots of different symptoms that follow along with that. And we can sort of see at the branching of different blood vessels how this can really impede blood flow. So a lot of different symptoms, and particularly if we go back to the notes looking at how we talked about the development of this disease and the phenotypic pattern, right? And how that phenotypic pattern can be different in each different individual. But here's some of the phenotypes as a result of sickle cell: kidney failure, heart failure, spleen damage, impaired mental function, paralysis, and then on the other side, other effects: pain, fever, joint problems, physical weakness, right? Fatigue, that sort of thing. All right. So back to our mutations. All right, and just going through how mutations occur. Excuse me. So one of the primary ways in which mutations occur is simply errors in DNA replication. And remember I said that this does happen, although we have spell checker enzymes that ensure the right nucleotides are paired up. Sometimes those are missed, and errors in DNA replication lead to mutations. It's extremely rare because of the effectiveness of DNA polymerase proofreading the strand, and errors are taken out. but they do occur. I think if I recall, I mentioned something like 1 in 10 billion or something like that. So that's pretty rare. But they do lead ultimately to things like this, where a mistake has been made in the genetic code, which leads to an altered protein. Another common way in which mutations occur is through substances called mutagens. All right, so these are substances in our environments, and they can come in many different forms. They can be chemicals that we perhaps breathe in through the atmosphere, or that we ingest through the food that we eat. They can also be things like radiation that we're exposed through, UV from the sun, radiation from an X-ray, or other low-level radiation, even trying to understand the effects of our radio waves that we use for texting, our phones, listening to your speakers via Bluetooth. All these are low-level types of radiation, which are supposed to not be damaging to our DNA, as opposed to higher level, higher energy radiation like UV light, X-rays, that sort of thing. But those things can damage our DNA. And again, we do have the ability of DNA repair enzymes that monitor and repair these changes that can occur, but ultimately mutagens can lead to changes in our DNA as well. Third one here, referred to as transposons, all right? And transposon literally means jumping genes. And these are where sections of our DNA genes can move to new locations within the chromosome. So they move to new locations on the chromosome, and they can disrupt sequences. So disrupting specific sequences, and particularly in the terms of important sequences for specific proteins. Lastly, we have discussed this term before when we talked about cancer and did our cancer lab, but similar in the line of mutagens are carcinogens. So of course, they're kind of the same thing, except carcinogens are specifically affecting those checkpoint proteins of the cell cycle. So that leads to the uncontrolled cell division. So whereas mutagens could be just any substance that influence any gene within our body, versus carcinogens, which they tend to specifically impact those proteins that ultimately lead to the development of cancer. Yeah, so basically any of the above lead to changes in the nucleotides of the genetic code, and they fall under a couple of different categories in terms of how we identify them. The most serious is what we refer to as a frame shift, all right? And so a frame shift is the result, it may also be referred to in a textbook as an addition or a deletion. And so if you can look over here in this image, here we've got our normal DNA and the mRNA that results, and the amino acids that would result from those mRNA codons. So in the case of an addition or a deletion, all right, an addition or a deletion, so we may also call that an insertion, so addition or insertion. What happens here is you're adding an extra nucleotide where it doesn't belong. So in the case of an insertion, if you look back up here, we have CTT. We've added another C, making CCT, which is then pushing this T over into the next set of codons, right? And what happens here is we now have a frame shift in the reading of our groups of three. So if I go back up here, right? Here we're reading in groups of three. So once that addition or insertion occurs, we can see the first two amino acids are normal, but at the location of the insertion, glutamate now becomes glycine, so this amino acid is altered. And because this nucleotide now gets shifted over in the reading frame, it becomes part of the next one. Every following amino acid from here on is going to be disrupted and affected. And that goes for a deletion as well. Hence why we call these frame shifts. So the ribosome is now reading a different group of codons from the point of the insertion or the deletion. So in this case, you can see we've removed the base here. So this one's being taken out, and then that shifts the reading frame. So from that point on, so this tyrosine becomes threonine, glutamate becomes lysine, and again, every amino acid from here on is affected. So it's definitely one of the more worst case scenarios in the mutations, a frame shift, so additions or deletions. And then we have what are called point mutations, and point mutations are often also referred to as a substitution. So in this case, you've just replaced, right, one nucleotide for another. So it doesn't lead to a frame shift, it only messes up the one set of codons that the substitution has occurred in. So here we have CTT, so they've changed this C for a T, which changes the codons to an A, and now instead of glutamate, we have lysine, but it's the only amino acid affected. From here on out, everything else will be normal. We sometimes refer to these as silent mutations, sometimes nonsense or missense mutations, because if we remember back to the codons, the table of codons, so if you remember this, we can see how sometimes an amino acid will have more than one possible set of codons. So for example, alanine could be GCU, it could be GCC, it could be GCA, or it could be GCG. Any one of these will lead to alanine. So sometimes when a mutation has occurred and it replaces the letter, it doesn't end up changing the amino acid at all, sometimes. And so in that scenario, it really becomes what we call a silent mutation or a nonsense because it doesn't disrupt the protein synthesis at all. And that's it. Pretty straightforward. I really do want to encourage you. I'll just bring it up, the video to watch. One moment. So if you look under videos, protein synthesis, transcription, translation, you open up that book. You will see down towards the bottom, as I mentioned earlier, lots of videos in here worthwhile watching to give you the visuals. But this one here on mutations, really good to look at gene mutations and how sometimes a single letter in millions and millions of letters can have quite drastic consequences. All right, enjoy the rest of your weekend, and we'll see you Monday.